I've been involved a lot in vaccine development in the past like 30 years, and I can tell you, and I've also been involved in a lot of first-in-human clinical studies, and I can tell you, you do not want to be one of the first to take this vaccine. There's almost certainly going to be unexpected side effects. There's going to be learning experiences. Doctors are going to realize they make mistakes sometimes that they didn't know from lack of experience. There's all kinds of things that are going to happen. I'm actually telling everybody, social distancing right now is probably better, and wait until about April. That's what I'm saying.
Wow.
About April before you actually take it. Let's see how many people, and then I don't know how many immunologists there are on this call, but
0 at the moment.
we have the VD and J regions of our chromosomes that kind of tell us what kind of immune responses we can have, and everybody's different. So even though in maybe the studies they've done so far, they haven't seen anybody have severe immune reactions to the vaccine, there's probably going to be some. And so I want to see, is it going to be 1 in 100 million, or is it 1 in 100,000, or 1 in 100 before I actually get vaccinated? As I think a lot of us are, we're in the very high risk group, and it would be good to get vaccinated, but I think it's also risky to get vaccinated at the moment.
Risk of getting.
And don't take the AstraZeneca one. That's a really dumb one. Okay, the Pfizer and Moderna ones are actually quite. They're probably the best you can possibly do. There'd be some other ones that could even be better coming along later. And I know Life Extension Foundation in Florida is now put together a vaccine that's actually pretty good. And I don't know what they're going to do. I don't know how they're going to make it available to people, and I think it's going to be super expensive. But
are the chances of getting COVID worse than getting the vaccine?
Say that again? I didn't hear you.
Are the chances of getting COVID and having side effects of that worse than getting a vaccine with having side effects?
Well, the effects are worse, but the chances are based on you. I mean, like what's your social distancing exercises? I'm extreme at the social distancing, so I'm not getting the vaccine. But if I am
halfway your ultra marathon, you don't meet anyone.
Yeah, well, yeah, you do. That's why I don't go to ultra marathons now. But if you go grocery shopping and stuff like that, then I would probably get a vaccine.
Sue, did you want to say something? You're on mute.
Yeah, I have a question. Why not the AstraZeneca vaccine? Because I have a mutual colleague that's actually working on it right now. And I mean, my son received the AstraZeneca. So he's out in San Diego right now.
It's using the adenovirus, okay? And it's a DNA virus, and the adenovirus is already known for being very harmful in gene therapy studies, where the adenovirus causes an extreme immune response in people, and so a lot of cytotoxic T cell responses occur. So what happens is that your immune system starts attacking not the gene therapy or not the vaccine, but it will start attacking, let's say, cells that have engulfed the vaccine or engulfed the adenovirus. Okay, because first the adenovirus has to produce the vaccine, and that requires getting into cells. And so the immune responses are tremendous. Now, they picked adenovirus because at least it's not as bad as some of the earlier gene therapy methods like lentivirus that actually integrated into the chromosomes, and that caused high cancer risks. So and then like when they did the bubble boy gene therapy stuff, I want to say 25 years ago, they treated 7 boys and 5 of them got cancer just because of the gene therapy. It's just the delivery method. But I would say the RNA, using an RNA or protein, so putting RNA into your cells or blood, or putting protein into your blood is the safest way to go, because it's less likely to cause immune responses and mutations that could cause cancer. But I'm not certain why AstraZeneca chose to go the adenovirus, except they thought they could get it done quicker. So I don't know. I always get myself in trouble. So now you're going to go tell your friend I said this.
No, I'm not going to say a word. And maybe they pivoted. I don't know.
Yeah, I normally don't badmouth people or things like that, but I just on this.
I mean, I'm right down the street from the headquarters. So.
Oh yeah.
Corey?
So was AstraZeneca the one that they were they the ones that partnered with Oxford?
I can't,
Yeah.
I don't remember
Okay.
that.
Okay.
I
Well,
think they were. I think so.
yeah, I just remember reading something about their meeting with the FDA back in September where they had like halted their trial at 9 o'clock in the morning and then didn't bother to say anything. They had a big long meeting that afternoon with the FDA and didn't bother to tell the FDA that, oh, by the way, we halted our trial. And then it came out in the Wall Street Journal at 5 o'clock that day. And I don't think the FDA was very happy with that. But my question was, you mentioned, you know, the differences in the virus. My understanding is if you're getting the vaccine, you just show up and you get the vaccine that wherever you go has. It's not like you can go down to your local drugstore and say, okay, I want a vaccine, but give me this one and not that one. It's just kind of like, here's your vaccine, you get what you get.
And that's where, like when I go to Walgreens for vaccines, I can choose.
You can.
They don't give you the option. You have to tell them, you know.
Well, like the flu vaccine, there's one for children, there's one for adults, and then there's one for seniors. So you would probably want you know, the 13 valent vaccine, right? And if you're going to get, I don't know. I think that's what it was.
Yeah, I mean, I guess you just figure out wherever you're going to go, figure out what vaccine they're given, and make sure you go to make sure you go someplace that's given the vaccine that you
Interesting.
want.
I hadn't given that any thought. You know, I'll put everyone back in the peanut gallery to give Bill a chance to tell us what he prepared today. I've kept Luke on this long because everyone enjoyed his sweater. Let's just enjoy the holiday moment that he embodies. And now we can start our show. Bill, you paid me a terrific compliment when you said that mine was your favorite conference ever. I'm really very, very flattered. But I will tell you in return that among all of the presentations that have traipsed across my stage, yours is among the most viewed videos ever.
Oh, really?
There's just two that were more. One of them was about Theranos. That
Yes.
got tens of thousands of views.
Liz, what's her
One
name?
was my buddy Mike Sperduti, who gave a rousing session about sales, and that was very timely. But you and Liz tag teaming on the effects of aging is right up there. And so for those of you who haven't met Bill before, he's one smart dude. He's a doctor, just like Jill Biden. And his good looks belie his age, which he just told us earlier. He's going to be 70 within the next 12 months. And well, I don't know. Are you going to do an ultra marathon? Because you said you kind of gave up on him. Is it just to thrash the existing 70-year-old record for running 100 miles? Is that the thrill of it?
No, I can't wait to get back into running ultra marathons at least once a month, just as for the adventure. So the breaking the world record is going to be just something that's just going to happen because it happens every time. You know, like I'm 69 years old. I haven't run last ultra marathon I ran was just at the end of 2019, and I finished second overall and broke the world record for a 70-year-old. Even though I wasn't 70 yet, but I broke the world record for a 70-year-old, and thought it was a piece of cake. So I think I'm going to be okay. I just gotta.
What is the present record for? How long was the ultra marathon?
100 miles.
And how long did it take you to run this distance?
It took me 20 hours, and it was the world record is 22 hours.
Wow. I'm sure you have. I haven't done in my head. What is that? How many miles you ran an hour?
That's a 12-minute mile pace.
God help us. Wow.
Yeah. Well, it's actually,
your body's not ready to just absolutely fall apart and need to be regenerated for an entire week before you can function again.
no, it's if you like, I have a lot of experience with inflammation. I used to be head of inflammation for the Burlex Biosciences Corporation, and the one thing I've learned is that inflammation can occur in joints and things like that if you don't run often enough. But if you run every day, your body is trained to recognize that as nothing foreign and don't have an immune response against it. So the people that, like, I have an identical twin brother, okay? He's suffering from arthritis in his knees. He's talking about getting his knees replaced. His knees creak when they bend. He's asking me for advice all the time. But he doesn't run like I do. He lives on a boat, hangs out on his boat all day long, does no exercise whatsoever. And that's been going on for, like, I want to say 25 years ago, we have photos of each other and we can't tell each other apart. Okay. Now it's like an amazing difference, and his health has fallen apart. He looks 10 years older than me now. And I think it's just it's not the exercising, it's the fact that it's consistent exercising that is really the secret. Okay. And it keeps your joints lubricated so that joints don't go bad if you run all the time. But if you run every 2 weeks, you know, your joints get really tight, and then you go out and run, and that causes a lot of stress on your knees because you've gone 2 weeks without running. And then these people that run marathon, you know, they run maybe once a month, they go run a marathon, they do really well, crossing the finish line, throwing up on their hands and knees. They are inducing tremendous inflammation. I've done a few studies where I've measured like electrolytes and C-reactive protein and things like that before, during, and after ultra marathons, and it's amazing. My C-reactive protein remains undetectable. Okay, whereas some of them skyrocket up into like the teens or higher than that for C-reactive protein. It's like, I think that the myths, the hearsays and stuff like that have led people a lot in the wrong direction. I'm genetically not disposed to not having joint problems, and that's my identical twin brother is the same genetics as me. That's proof of that. But I don't have any joint problems. You know, I've been running since I was 10 years old. I competed my first race when I was 10, and I've been just addicted to the sport ever since. And I've run hundreds of marathons and probably 120 ultra marathons. It's something I should be a cripple if all the theories were true.
Somebody asked, what is your average daily distance?
Well, it depends. If I've got a 100-mile race coming up, it's really serious, then I'll average like 10 miles a day. I won't do, I actually have a routine of like I do 5 miles on Monday, 8 miles on Tuesday, 15 on Wednesday, drop to 5 on Friday, do nothing on, drop to 5 on Thursday, then do nothing on Friday. Saturday, I go out, run 25 miles, and Sunday I do 15 miles. If you add that up, that's 72 miles a week, so a little over 10 miles a day. But when I'm not, then I cut that in half. Okay, so and then I taper for 2 weeks. I essentially don't do any running for 2 weeks before a race, and then and no exercise. But if I'm not racing, I typically do like average about 5 miles a day, and sometimes I have a really hard time keeping that down because once I get started, it's hard, as I said, it's hard to turn around.
I think my audience should set back their clocks because Bill is fascinating and we haven't even started on the topic. I have one more question. They're asking, Joanne wants to know the difference between your and your brother's diets.
Well, his diet was terrible, but in the last like maybe 2 or 3 years, it's become identical to mine, which is I'm completely vegan. I don't do any oils. I'm a big fan of Dr. Caldwell Esselstyn and Dr. Amy Doneen. Caldwell Esselstyn is from the Cleveland Clinic. Amy Doneen's got a clinic up in Spokane, Washington. They've both published books. With my background in inflammation and stuff like that, I believe they've nailed it. They've got it exactly right. So that's why like I don't do any oils. I don't even cook in oil. Okay, I don't vegan. This is true for my brother too. Trying to avoid arachidonic acid is the key thing that's in all our meats and vegetables and meats and dairy products. So that's my. I'm trying to think if there's anything else I do special in my diet. I stay away from nuts and seeds, things like that, mostly because of the oils in them. And Caldwell Esselstyn recommends it. Yeah, I'm sure there's other things, but I do take a lot of supplements, and I drink a lot of protein shakes because, as you know, plants do have proteins, but they don't have enough of them, and they don't have the right balance of amino acids. Like threonine is often totally absent from a lot of plant proteins. So I get vegan protein shakes that sometimes I supplement with my own amino acids because sometimes they're deficient. But a lot of people think, God, you go to all this trouble. I kind of think it's fun. It's been an obsession of mine my whole life to just try to find ways to stay healthy and have fun doing it.
We'll have our thing. I won't admit to mine right now, but last comment from my very Italian friend, Andre Domino. He said, "What? No Italian food?"
Well, it depends. I do eat lots of Italian food, but it depends on how it's cooked. Like the problem with most pastas is it does have oil. It's a lot of oil in it. And then a lot of Italian foods do have oil in, you get a bowl of something and you see more oil than you see anything else in it.
He's in New Jersey. There's no threat. You don't have to go to his house. Bill,
Can
tell us about.
Excuse me for 30 seconds. My assistant's asking me to sign something.
Well, that's funny. I was just about making the segue into tell us about telomeres and the like, and I'm looking at his painting.
Okay.
Happy holidays, everyone.
I'm sorry.
He's back.
So
Just about what you know so well.
okay, well, I've got a PowerPoint presentation. Let me go ahead and bring that up.
Okay.
Why some other screens are popping up in front of me when they shouldn't be, I don't know. Okay, so I'm going to start off with trying to answer the question that seems like everybody always asks me at talks, and that's why am I so obsessed with curing aging? There's a lot of benefits to society. I think in curing aging, things are bad now. If there was a cure for aging in 100 years from now, nobody would ever vote to ban it just because there's so much good that could come out of it. But I personally am into this is because I think there's a lot of great things that are going to happen in the future. People might recognize this picture from Star Trek First Contact when humans first meet aliens, make first contact with aliens from outer space. When that happens, I want to be there. That's it. I am obsessed with living. I love living. I think it's going to be, I could never imagine not living. I don't want living to ever become less lovable because of aging. And that's for me or anybody, and, a lot of people take living for granted, and it's the greatest thing that ever happened to us. It's like, I'm not going to just let it get thrown away. Okay. So that's me. And the reason I just said it is because a lot of people, people have stood up in conferences and just yelled at me and said, "What, are you afraid of dying?" And I'm not afraid of dying at all. I wouldn't be running ultra marathons in the middle of the night when running into mountain lions and stuff on occasion if I was afraid of dying. It's just that I don't want to miss out. This fear of missing out, okay, is why I am so obsessed with this whole thing of curing aging. I've been interested since I've been 10 years old, and because my father put the seed in my idea, had to cure aging, but I've been 100% focused on this for the last 30, 40 years. So I do run and I own a medical research company called Sierra Sciences in Reno, Nevada. We're a 10,000 square foot research facility. And when I say 10,000 square feet, we don't have any business sections. It's all labs, okay? It's all laboratory. We have everything that you could possibly imagine ever needing to do any research on aging. That's what we do. Okay, so I founded this company about 21 years ago now, and before then I was working at Geron Corporation, where I first got really into what I'm going to talk about today. But our goal is to cure aging, and this thing, there's a lot of people out there saying they've already got cures for aging and they're working on it, and this is one of the problems with biomarkers of aging. A lot of people are always talking about all of these great things like NAD levels or methylation on your chromosomes, and then I can't think of any reason why anybody would really care about that, except if they're trying to develop a product that they can advertise saying, "I have a cure for aging," because they'll say, "Oh, look, I figured out a way to reverse the methylation pattern," or "I figured out a way to change the telomere lengths," or "I figured out a way to increase your NAD levels. Therefore, because it's a biomarker of aging, I've reversed it. I reversed aging." And this is so there's so much, I'm going to say, crap in the industry right now. People selling products that don't really work. I'm obsessed with curing my own aging, so I'm not going to participate in the business of trying to mislead people into something that doesn't really work. But that's what biomarkers of aging do. But in reality, if you're a doctor and a patient comes and says, I want to slow my aging down, this is what they mean. They want to, this is Betty White, okay? Betty White, 25 and 95. Okay, I think maybe she was actually 90 there, but it's like, this is what people are talking about. And I don't know, what biomarkers are we referring to there? People don't go into your office and say, I am dying to get my NAD levels higher, or I'm dying to get my methylation patterns changed in my DNA. They don't go, that's not what they want.
They don't know the language. I know I don't know half the things you're talking about there.
Oh, really? Well, that's so much in the press lately. Maybe I'm a little biased in what I read, but there is so much in the press right now. Like, if you ever heard about there's hyperbaric chambers right now are being advertised.
What are those?
Okay, well, they're now saying they cure aging, and the papers that they publish is full of crap. I can find every flaw in the world on it, and I've been interviewed by 2 news reporters, and I just went through all the details. But that paper led 2 weeks later to them starting a company in Dubai to now start providing hyperbaric chambers to cure aging, and everybody believes it. But it's not true. It's nobody. I always say, well, okay, so we want to slow this down, but it's really hard to measure slowing. So my focus is I want to reverse aging. I call it the cure for aging because I'm not a semantics expert, but and people criticize me for saying cure for aging all the time, but this is what I want to do. I want to turn us this way. And I don't believe anybody has ever has ever come up with a way to cure aging if all they show is some biomarkers got aging. They have to do this. They have to show that by visual, we can tell that Betty White's gone from old to young. And I don't think there's anybody here listening to this right now that doesn't know which photo of Betty White was taken first. And if you want to provide me with a list of biomarkers that you just measured to be able to tell that, let me know. Let me know what those are, because I'd like to include them in my list. But right now, I don't know exactly what it is. Okay, there's wrinkles, there's other things going on, but clearly this is what we want to reverse, and that's what I'm focused on. So my biomarker is the Betty White test, I call this. I've been speaking at conferences for this, and people are getting up. I don't want to mention names, but people they get up and talk about how they cured aging all the time, and I'll just say, does your cure pass the Betty White test? And the answer is always no, because nobody's ever been able to do this really, at least in humans.
Let me understand. So
yeah,
I've been thinking that that you want to slow aging or stop aging, but you're saying you'd actually like to give me back 10 years as well?
Yeah, that is the goal. That's been my mission all my life. Okay, is to figure out how to reverse aging, and but slowing aging and then stopping aging are going to come first. Okay, so as I'll describe, I think we already have some things that will slow down the aging process. We don't have anything that was, well, we don't have anything that's been shown to stop or reverse aging in humans, but we do have some things that have succeeded in some animals.
If it's not the next slide, could I ask you to advance to the one? I remember your shoelace example. That's the way I kind of hold on to what you taught me. Can you show folks that if it's in your presentation at all?
Yes, I'm gonna come to that. I'm not gonna. I took the shoelace picture out, but I just want to say that my company, all we do is discovery research. We don't sell anything. We are just doing the research. Okay, our major goal is to discover a pill that will pass the Betty White test, and our funding is actually right now all from go-to-market partners. And so that's going to be actually one of the things I'm talking about. Since I'm talking to a medical device group, it's pretty hard to figure out how I fit into medical devices, but I think I'm going to have suggest some ways that there's going to be overlap here. So what we do is we discover products, and most of these are by accident. Okay, so we're just trying to do research, but a lot of times we end up discovering something that somebody could market if they wanted to, but we do not want to do marketing. We want to focus on just the research. So we will license these things to others that come along and say they would like to get involved with us and market products. So we license it to them. We just ask for like a 10% of gross profits in return. Then we use that to fund our research. That's how all of our research funding is done. I used to have investors. I raised $30 million in the first 10 years from investors. But the problem was that the investors were always interested in return on investment, of course, but they would steer us away from what we really needed to be doing, and then start talking about, hey, look, we're being very successful. Let's sell the company, And that just defeated everything I wanted to do. So I ended up figuring out a way to buy out all my investors. And ever since then, I've been getting my funding from go-to-market partners. And that's our business model now. So let me just quickly who I am. I'm the founder, president, CEO of Sierra Sciences. I founded the company in 1999. By background, I'm a medical researcher. I've had a lot of success at inventing things that are products on the market now. Okay, and I'm one of the inventors of human growth hormone and tissue plasminogen activator, erythropoietin. I don't know if you guys know what these are, but like erythropoietin is what Lance Armstrong used to cheat, but it's something that helps people boost red blood cells. I've been involved heavily in a lot of these different things. These are things that I actually played major roles in, that they would have never been on the market if I hadn't come in and done what others couldn't do. So I'm actually really good at the lab bench. That's why I don't want to get into marketing. I want to stay at the lab bench and get some of these things done. I've beta-seron, the very first drug ever for treating multiple sclerosis. I'm one of the inventors of that. And then telomerase, which is what I'm working on now. For the last 30 years, I've been 100% focused on curing aging. And the toughest part has been finding the funding. When I had investors, I was getting the money I need. I was spending about $1 million a month on my research. When I got away from my investors, that's dropped to maybe like $100,000 a month. At least it keeps us going. We just can't get the research done at the pace that we used to, but we couldn't get it done before anyway because they kept sending us off in different directions. Education, my PhD, and I never call myself doctor. The only time you ever hear anybody say doctor is when I get introduced. So my PhD is in molecular genetics and population genetics. Population genetics is mostly the study of evolution. But molecular genetics is the study of how genes work and stuff, which I think those two come hand in hand in my aging research. My bachelor's degree is in biology and psychology, so I actually am quite knowledgeable about the psychology of aging too. And then I minored in statistical theory, which actually came in really handy because it makes me really good at being able to read other people's papers and find out how dumb they are sometimes. You know, this wrong things they say, the wrong conclusions they reach. Sometimes they reach these wrong conclusions intentionally, sometimes unintentionally. But sometimes they're trying to market a product, and so they'll turn a blind eye to the fact that they did the study incorrectly. I'll catch that. I've been in biotech for 39 years now. A lot of my research has been inflammation, cancer, and heart disease. In the inflammation and cancer, actually, I never got any products on the market, but I did a lot of discoveries of a lot of different treatments for cancer and inflammation that got well publicized. They just never turned into products. I was National Inventor of the Year, second place in 1997 for my cancer research. First place that year went to the people that discovered the HIV protease inhibitor. And then during the press conference, all they did was rave about the work we did. I led that team that did the cancer research. Okay, so I'm also an ultra marathon runner, as we've already discussed a little bit before talking. I'm featured in 2 documentaries, one called The Immortalist, really good documentary, almost won an Oscar. And it's mostly about me and my research. It also includes Aubrey de Grey, who is included because he's the anti-me. I guess he's the opposite of me, and they wanted to show contrast. So he and I are both in this. But I'm also in a movie called The High, and this is a crazy movie where I ran 138 miles through the Himalayas of northern India at 18,000 feet elevation, nonstop. And so they filmed me doing this, and it became a documentary.
You know, I did see The Immortalist. I'm thinking I saw it maybe on Netflix.
Yeah, it's on Netflix, and both these are on Netflix and everything.
But why do you call Aubrey the anti-you? I think if he was also looking to.
Aubrey and I are very good friends, and I thought, oh, I shouldn't have said that the way I said it, because he and I are very good friends. We're extremely passionate about curing aging, but our diets are different, our exercise things are different, our lifestyles are different.
Your lifestyles are different.
People wonder all the time, how can Aubrey and I be friends? It's because of our passion for curing aging. He's the real McCoy. He is not trying to just make a product. He really wants to get aging cured.
How old is he?
He's like 10 years younger than me. I remember when we did the Immortalist, he had just turned 50 during that. So he's probably, no, I guess he's more than 10 years younger than me. But
Okay. So where yours is very laboratory specific, he's very what?
well, he's really not. He's got a PhD. It's mostly, but it's not anything to do with biology and medical. I think it was an honorary PhD. His background's in computers, but he's more somebody that spent a lot of time just really studying and reading everything there was to be read about aging, and then putting together theories, which I thought he did a really good job on, to try to then get people to start doing research in those areas. Now, so when I first met him, and he and I debated one time on Channel 4 News. That was actually the first time I ever met him, and it never got shown on TV because when it came my turn to do the rebuttal, I said, "Hey, I agree with everything Aubrey just said," and then Aubrey said, "Hey, I agree with everything Bill just said." So there was no rebuttals and never got filmed. But he's now I forgot the question suddenly. But he's
You originally regrettably painted him as the anti you, and I was
yeah, but he's not. He's it's just the lifestyle differences. Okay, but for some reason the producers and documentaries wanted to show that somebody really does have a cure for aging, and somebody is just trying to find something to do to get a cure for aging. Okay, and so they were contrasting us in that way. But Aubrey's been really good at trying to get scientists to start doing some of the things that he's put together, which I thought were really good. I think but I don't think they're as important as what we're doing, and I'll explain that in an upcoming slide too.
he looks more like a scientist than you.
He's more become a figurehead. He speaks and stuff like that, but he doesn't do any work at the lab bench. Okay, so I'm author of 2 books, Curing Aging and Lengthening Telomeres. I'll talk a little bit about those too, but here's the bottom line. I believe the kingpin of aging is the telomere, okay? And I believe that this is true for aging and health in primates, including humans, dogs, cats, horses, sheep, pig, and deer. We have not found any other animal that ages by telomere or has declining health due to telomere shortening. And that includes mice and all other rodents. Okay, so a lot of times you hear about aging research being done on mice, and I always argue, mice are not a really good model for aging in humans because mice age by an entirely different process than humans do. Bottom line is, I don't believe, I think there's aging is like multiple sticks of dynamite burning inside of our cells. We have a lot of things causing us to age, but no matter what else we do, nothing's going to stop the aging process unless we also stop the telomere shortening. And that's a major clock. That one thing I'm going to talk is an absolute, and there's nothing, no theory of aging that's more absolute than telomere shortening in controlling our health and aging. So now back to your question.
How far have you gotten in convincing the medical and health communities about telomere shortening as the metric by which aging research can be reasonably done?
I think it's pretty good now. I think it's there's still a lot of people, including at least one Nobel Prize winner, that argues that it has nothing to do with aging. But it's like I'm really good at what is called meta-analysis of scientific peer-reviewed studies, and I can actually, like if you went to my website, you could click on
On the Sierra one?
picture of sierrasci.com, sierrasciences.com. Click on a picture of an old truck, and you'll see a video there where I actually do kind of attack a lot of people who are arguing that telomeres don't cause aging, and in fact, they actually lengthen them is going to cause cancer. And I went through an entire meta-analysis to show that there's no data whatsoever to support what they say, but there's tons of data that argue the exact opposite. And when I say there's no data, there is some data, and you can find anything that you wish to be true in even PubMed. Okay, so if you want to find out if like drinking alcohol gives some people green horns on their head, you'll find a paper that says they do. Even though it's peer reviewed, a lot of papers get through. But you got to do meta-analysis, which means reading all the papers and then being able to analyze each of them. Having a background in statistical theory helps a lot, but actually having hands-on experience of actually doing the same experiments that they published and knowing the kind of things that can happen that can go wrong and stuff like that makes it really good, easy for me to be able to do meta-analyses. But I've done a really good job, I think, in my presentations of convincing just about everybody that this is, you said the metric of aging. I will say it's the cause of aging, but I would say our ability to measure telomere lengths is so terrible that I actually encourage people, don't waste your time measuring your telomeres. Okay, most of the companies that are providing that right now are probably just doing it for the money and don't even believe the results themselves. And I've had people test this by sending their blood samples in 5 different tubes with different names on it all at the same time and getting 5 entirely different results. That's because telomere length measurement is extremely difficult to do, but I'm going to be talking about that here a little bit in a moment because that is one of the.
Jan, you had a comment?
Oh, I'm just saying I've heard a lot of what he's saying and from everything I've seen the telomere, I can't even say the word, telomere shortening is something that everybody worries about, and I'd like to find out if he knows how to lengthen them. So I'm
Sorry. I expect that at a medical device conference there might be at least 1 or 2 people that doesn't know some of the background stuff. So I'll get to that in a minute. But let me first just describe what a telomere is. Get this. Someday I got to get this picture changed to something else. But the artist that made this decided to make a half-naked man. And
yeah, that's sort of weird.
Like pass this.
This started like 8 or 10 years ago, right? The telomere stuff.
Oh, not at all. I began my research 30 years ago.
Yeah, well, it's hitting the newsstands and information and stuff a lot more in the last 10 years, I thought. Might have started earlier.
The more people start hearing about it, the more people start thinking, oh, I could make a buck by saying something and getting people to come and buy things from me. And so it gets in the news a lot. But really, nothing has really happened except for research so far. And I'll explain that in a minute. Like Luke just came on. Did he have a question too? Or?
Yeah, of course. Don't stem cells start off with new telomeres?
Human embryonic stem cells do, and induce
All the others
pluripotent
don't?
cells do, but not. That's a myth. That's a hearsay. You'll never find a study at all where anybody's looked at telomerase or telomere lengthening in stem cells and actually shown that happens. In all cases, they shorten.
Okay, and I have another question. That is like 2 great biomarkers that show that you're aging. That's cartilage, like the nose and your ears. Do you want the reverse growing of them also?
I don't want to reverse development. Okay, so that's reversing development. So I make it
because
really
only some people get more handsome because they get a bigger ear or a bigger
absolutely.
nose.
I don't think they get more handsome from hair growing in their ears, but yeah, it sounded like that's what you said. I make it really clear that we are only focused on reversing aging, not development. Okay, so most of the people working in the developmental field will say that we pretty much finish 90% of our development by the time we're 25. So I'm thinking that we're going to reverse our aging to 25 if we do it at all, if it's possible. But not, somebody's not going to become younger than 25, okay, because that would be essentially reversing development then. Let me just go really quick here. So we zoom in on human being. The first see that humans are made up of cells. We've got 100 million cells, 100 trillion cells. Some people say 30 trillion cells, but that's pretty close to 100 trillion. And almost every theory on aging is saying that we age because our cells age. So my research is mostly on trying to figure out ways of keeping our cells from aging. So in my company, we don't work on anything bigger than a human cell. If we want to do testing on something else, we provide it to other labs to do mouse studies or human studies and stuff. But every cell contains a nucleus, and inside the nucleus are found our chromosomes. And there's a top arm and bottom arm here in this chromosome, and each arm contains this long DNA molecule of beads on a string. The beads are called bases, but they're about 100 million bases in length on average. And that's the shoelace analogy. So think of this long DNA molecule that's inside each of there's 2 arms. So each arm has the identical DNA molecule. It's all coiled up like a slinky inside this chromosome. But if you unravel it, it's a long shoelace essentially. And by the way, these beads, the sequence of these beads are what tells us what our hair color and eye color are and other things. But the genes are there. But the shoelace, have these caps at the very ends of your shoelaces called aglets. And these aglets protect your shoelace, okay? And so our chromosomes, the ends of our chromosome DNA, the DNAs in our chromosome, have the same thing shown here in yellow. They're called telomeres. The telomeres are the aglets of our chromosomes. Okay, they protect our chromosomes from all kinds of things, but they do play some additional roles too. I'm going to zoom in now on one of the telomeres, and the telomere is about 100 million, telomere is about 15,000 bases in length, so it's a pretty small component of the whole chromosome. It's about 15,000 bases, but that's only true when we're actually first conceived. When we're a single cell embryo, I'm showing more than a single cell embryo here because the artist misunderstood what I said. But when we're a single cell embryo, our telomeres are about 15,000 bases. But when our cells divide, this is the problem. And I got some really good slides on, but I'm not going to show them today on why this is. But every time our cells divide, our telomeres get a little shorter and shorter and shorter. So each and every time our cells divide, our telomeres get shorter. There's so much cell division going from a single cell embryo to a newborn baby that by the time we're born, our telomeres are already shortened down to 10,000 bases. And that's okay, because that's like cutting 1/3 of the aglet off your shoelace. Your shoelace is still perfectly fine. But the problem doesn't end here. We still have a lot of growing up to do, lots of cell division, lots of wounds to heal, lots of immune functions to fight. And so our telomeres get shorter and shorter and shorter. And when our telomeres get down to 5,000 bases, our cells lose the ability to function and we die of old age. That is what's happening. This is a fact. This is not a theory anymore. This is accepted by anybody who works with human cells in petri dishes, especially in laboratories, knows that when human cells grow and they divide, the telomeres get shorter. And when the telomeres get down to 5,000 bases, that's it. The cells don't grow anymore. They go into a phase called senescence, also referred to as the Hayflick limit, and our cells die. And that's what happens when we get old. Right? When we measure telomeres in people in old age and stuff like that, we find their telomeres are right at this critical threshold of 5,000 bases. Let me go over that again. We're conceived at 15,000 bases. We're born at 10,000 bases, and we die of old age at 5,000 bases. This is, again, not a theory. This is a fact. And it's really the only clock of aging that's ever been discovered. Every other clock that you hear about like methylation, NAD, sirtuins, there's a whole bunch of them, different things. Now, there's IgG glycosylation has been a good measurement of aging. All these things are actually controlled by the telomeres, and we know that from in vitro studies. So telomeres, I say, are the kingpin.
And let me stop you there.
Yes.
Interesting. You said controlled by the telomeres. And I'm looking at your animation here and thinking that the telomere is subjected to other things, like they are controlling the deterioration of the telomere. But you said controlled by the telomere, which makes me think that the telomere is doing.
Yeah, well,
is the catalyst.
There are things that do affect telomere length. And they're mostly lifestyle things. And I'll talk about that in a minute too. But there's really nothing that's controlling telomere length. The word deterioration in telomere length is a misnomer. Deterioration of telomeres is a very rare thing to happen. The shortening of telomeres is mostly caused by when a cell divides. And I have really good slides. I wish I had those here right now. I could bring them up if needed. But when a cell divides and everything inside that cell needs to be duplicated, that includes the chromosomes. And when the chromosome is duplicated or replicated, it's not replicated all the way to the very end for reasons that have nothing to do with deterioration or poor biology. It's just, it's like the analogy I usually use is a bricklayer making a new row of bricks on top of a brick wall. I have actually good cartoons showing this. And the bricklayer is backing up, putting a brick down, backing up, putting a brick down. And when the bricklayer reaches the end of the wall, he or she falls off the wall and can't put a brick in the last place. As a result, the new layer of bricks, new row of bricks is shorter than the previous row. So as a result, our telomeres get shorter, not because of deterioration, but because of the cell's lack of ability to duplicate the chromosome all the way to the very end. Every time
the rest of the chromosome duplicated and only the telomere is compromised?
Yeah, at the very, very tip. So
Why would that, I mean, theoretically, of course, this is way over my head, but theoretically, if everything else is being duplicated, why is it just the ends that are compromised?
Because of the machinery that's used to replicate DNA, okay? DNA polymerase, I do have, when I talk to scientific conferences, I do show all the Okazaki fragments and primases and things like that that are involved.
Way too much for me to handle.
It's the biology. It's just that when you get to the very end, the polymerase has no ability to actually replicate the last piece of DNA that it was attached to. Okay, as soon as it moves over to fill in the missing spot, it falls off. Okay, so the new chromosome is shorter. And
hey, if I could build on that, let's go with that as the foundation of your work. Is your thought that your science would help us split a cell and keep all the telomere, or
yes,
would we be able to build? Can we do things to grow our telomeres?
Yeah, originally when we first discovered the ways to fill in that gap, okay, that's all we were trying to do. But then back in the early 1990s, when we figured that out, we actually, when we looked at the telomere length, we found it got longer. Okay, so it was almost as if the bricklayer got wings and could fly out and start adding bricks longer than the previous row. And that's in the cartoons. I actually show the bricklayer become an angel and actually adding these bricks. So they actually
in the 90s you figured out how to do that?
Yeah.
And are we doing that? No.
Yeah, I'm a scientist. I can't understand why business people aren't jumping all over this. Okay, and it's just and everybody that has not been very good at marketing and things like that. But this is, I think, the greatest thing that ever got discovered in biology. And I'm sitting here trying to figure out why isn't it.
Is there perhaps years from now a pharmacological solution where I could, this is silly, I'm using baby language, eat telomeres and get them in my system and make them longer?
Yeah,
So creating a drug?
The way you just said it is wrong, but that's my goal, is to develop a pill follow that's going to make your telomeres long.
Okay, say it in not so, maybe give me a grammar school level of understanding how I would grow my telomere.
That's gonna come in the next slides.
All right.
Okay, the very next slide. So the next slide is lengthening telomeres. Okay, so I was saying that our cells lack the ability to lengthen all the way to the ends. But if that was completely 100% true, then we would have been extinct as a species by now, because our children were born with shorter telomeres than we have, and their children were born with shorter telomeres, and we wouldn't have any chromosomes left. And it turns out our reproductive cells don't have telomere shortening. Okay, this is what we learned in the late 80s, early 90s. And so my mission at that time was to figure out why not. Well, how is it that when our reproductive cells divide in petri dishes, when I say reproductive cells, I'm talking about primordial germ cells now, not actual sperm and eggs, but the cells that produce the sperm and eggs, called primordial germ cells. Why is it that they don't have telomere shortening? They're the only cells in our entire body that don't. Okay, they're also called human embryonic stem cells, and you can make them, and they're also called induced pluripotent stem cells when you make them out of other cells. But in these cells, telomeres don't shorten. So my team at Geron Corporation ended up discovering human telomerase. Okay, this is an enzyme. It's a separate enzyme, and in the bricklayer models, I use this as the angel with wings that's laying the bricks. This picture is a cartoon. The green squiggly thing is the DNA, shown as a double helix here. And telomerase is like a factory. It's an enzyme that actually binds to the very end of the chromosome and fills in that gap, makes it longer. And then we found out unexpectedly, of course, I always hoped it would be the case, that when we actually put telomerase into, put the enzyme into cells, the telomeres actually got longer than the previous chromosomes. So, which was a good thing, because even though there's some people that say that there's such a thing as having telomeres too long, that's not true. There's no data whatsoever saying that long telomeres are bad for you. So so we discovered this enzyme. It's very difficult. The enzyme's too big to just swallow and put into your cells or inject into your blood. To actually get cells to produce it, you have to, or to have it, you have to get them to produce it themselves. because they all have the gene for it. So we had to use some manipulations in the mid 1990s, gene therapy manipulations, to actually put the telomerase gene into cells and get these cells to start producing telomerase. When we did that, we found out that we reversed aging in human cells in vitro. So just when you grow cells, first thing we did, we published this in early 1990s, I think. We showed that we actually reversed aging in human cells grown in a petri dish. We showed that we had totally abolished what's called the Hayflick limit, which is something that everybody, it's senescence, cell senescence. We completely obliterated cell senescence by getting these cells to produce telomerase. And that made actually headline news back in the 1990s, and then everybody forgot about it. Okay, but we also, so at that time, the dumb thing was, it's frustrating. So I was at Geron Corporation at the time. They hired me just to go ahead and discover human telomerase. Because of my background with human growth hormone and other things like that, they thought I was the guy to do it. And I did it, and it only took me 3 months. But I also did what's called the antisense. Okay, I took the antisense of telomerase and put it into cancer cells and showed that I could kill those cancer cells by causing accelerated aging in those cancer cells. And it worked on every cancer cell, and it had 0 side effects on non-cancer cells. So Geron Corporation decided, they're investors, and this is why I don't like investors. Their investors had a meeting. I spoke at the meeting. They said, okay, so Bill, you've just in like the matter of a few weeks come up with a cure for aging and a cure for cancer at the same time. We can't do both. We have to focus on just one. And it was unanimously decided by the investors that there was a faster return on investment by focusing on cancer. I tried to argue that. I said the #1 cause of cancer is aging, and you are only going to cause slowing of cancer, but you're going to increase the rate that cancers are going to come back. But they ignored me. They turned to cancer. I stayed a little longer, started Sierra Sciences to continue the aging research. And then scientists that were still on board with me, that were still at Geron, did this next experiment. They took human skin, grew it on the backs of mice, and then they had old human skin and young human skin on separate mice, or sometimes on the same mouse. Then they sometimes treated with telomerase and sometimes didn't treat with telomerase. And they found out that when they treated old human skin grown on the back of a mouse with telomerase, it reversed aging in every measurement they can do. Okay, they saw wrinkles disappear. They saw the ability to blister disappear. They looked at 30 different biomarkers of aging. All of those got reversed. That was 2010 when that got published. No, 2000. I think it was 2000 when that got published. Again, it seems like the world's forgotten about that. And then we sent our technology to a lab in Harvard run by Dr. Ron DePinto, who was actually, I met him at a conference in Hawaii, and he wanted to prove that telomerase causes cancer. And I said, okay, well, here, let me give you everything you need to do that. He then engineered these mice so that they would produce telomerase. I mentioned before that mice don't age by telomere shortening, so we had to use engineered mice that did. He lengthened the telomeres and expected all these mice to get cancer, but the rate of cancer actually decreased, not increased, and all these mice became younger. Okay, so Diane Sawyer did a really good story on him. If you do a Google search for Diane Sawyer and Ron DePinto, you'll see the interview, which I sometimes show in my slides, where he called this a remarkable reversal of the aging process. And I don't know if you guys know who Dr. Ron DePinto is, but that kind of destroyed his career, because his career was to fight cancer at Harvard. And he was thinking telomerase was the cause of cancer, but it was
DEP.
So his name is DEPINHO. Just do Diane Sawyer and Ron DePinto.
December 2010.
Yeah, and he actually saw this remarkable reversal of the aging process. He'll say in the show, but he was trying to show that the mice were going to get cancer. So that kind of, like I said, destroyed his career. So he left Harvard and became the head of MD Anderson. Okay, to focus on cancer research there.
Do you mind if I have the video up? Do you mind if I show it?
Yeah, go ahead.
Okay,
but you might find you're using one that's from somebody that's trying to promote a product. And
no, I don't I don't think I am. Share screen. Yes. There. And I have the mute on.
In a cage around the corner.
Is this it?
Yep.
For some pioneering
About 2 minutes long.
wonder what does a fountain of youth for rodents reveal for humans? Here's Sharon Alfonsi reporting. In the movie Cocoon, it's a swimming pool that turns back the clock for senior citizens. But now researchers have found a way not just to stop, but reverse the aging process. The key is something called a telomere. We all have them. They're the tips or caps of your chromosomes, seen here in yellow. This is what it looks like in a young adult. But as you grow older, the telomeres become damaged and frayed, and as they stop working, we start aging, experiencing things like hearing and memory loss. Scientists took mice who were prematurely aged, added an enzyme, and essentially turned their telomeres back on. You can see it before the enzyme, after.
Their brain function improved, their fertility was restored. It was a remarkable reversal of the aging process.
Look at this picture. The mouse on the right has bad skin, gray hair, and is balding. But the one on the left had its telomeres hooked back on.
And you can see that essentially you now have a dark coat color, that the hair is restored, that the coat has a nice healthy sheen to it.
Even more dramatic, the change in brain size. This is before. The mice had 75% of a normal brain, like a patient with severe Alzheimer's. But after the telomeres were reactivated, the brain returns to normal size. As for humans, while it is just one factor, scientists now say by looking at our blood cells and measuring those telomeres, we can get a better idea of how well you'll age. The longer the telomere, the better the chances for a more graceful aging. But as for tinkering with them and turning back our aging process, researchers say we still have a long way to go. Sharon Alfonsi, ABC News, New York.
Yeah, so I don't know if you want to unshare your screen. I'm a scientist. I am not a good businessman. I am the first one to confess that. I don't understand why we don't have every investor in the world knocking on our doors trying to build on this thing. And I keep giving these talks, but one of the things that I try to do with these talks is trying to get people to help me. Okay, people. I mean, okay, so like Ron DePinto, he's a scientist. He became the head of MD Anderson as a result of all this. And but scientists don't make good business people, and so he got fired as head of MD Anderson earlier this year. It's like I don't know what he's doing now. Yeah,
because what? There were a speculation as to why he was fired.
I just don't know. I just know that every scientist I've ever known, that actually every good scientist, who's a remarkable scientist, every good scientist that ever went into business failed. Okay, that's why. I personally don't want to be president of my own company. I keep trying to bring in other people to be president, but they keep failing. And so I'm still just the president of my company. But we're all research. If I walked out the door behind me right now, I'd walk right into a lab where I would be doing science, because I have an accountant that I call a chief operating officer that takes care of everything, but she's got practically the smallest office in the building. So we're totally focused on science, and I'm trying to stay that way. But
you're a businessman. Help this guy.
Hey, Bill, that's interesting. You know, I used to come out to Geron years ago, but I had a question. Have you guys looked at any of the vectors, like the simplex viral vectors or the lentiviral for delivering this?
Yeah. Oh, yeah. No, we do all that. We have every one of those, and the adenovirus and the adeno-associated virus. The best one for research is lentivirus, and that's because its infectivity rate is so high, but it also integrates into the chromosomes and causes cancer.
Exactly. Well, that's why I was wondering it may actually help with delivering the telomerase, so you can reverse the process, right?
I developed a gene therapy which I think is the safest so far. Using adeno-associated virus. I constructed that. I built that 10 years ago now, and I've been trying to get somebody to do with it. I even licensed it to a company called Labella Gene Therapeutics 3 years ago to do clinical studies, and they never got their first patient treated. I just terminated the contract just about 2 months ago. So
we should talk.
Yeah.
You know, in my previous company, I had a company called Actus Biologics, and I was working on lentiviral vectors at that time. This was back in 2005 to 2011, and I don't know if you know Dr. Noriyuki Kashihara. So he and I were involved in this project together.
What's his first name? Because I know Akasha.
Noriyuki Kashihara. Nori Kashihara.
No, I don't.
He was the founder of the technology which was by Tokagen, used by Tokagen.
And what's their technology?
This was on adeno-associated virus.
Okay, so adeno-associated virus is really not a great virus. It's actually, it's the safest, but it like in our studies, we have to use 30,000 times as much virus as there are cells to get 50% of the cells infected. So its infectivity rate is low. So I first proposed gene therapy for curing Tay-Sachs disease in the late 1970s. I've been involved in that kind of stuff. And at the time, that was before lentivirus, but we were using retroviruses. Michael Bishop and David Baltimore both tried to recruit me to their labs to focus on developing gene therapy protocols to treat diseases like Tay-Sachs disease. But I was always concerned about the integration causing mutation. So I actually never actually, so I've done lentivirus, tremendous. In fact, a lot of the studies that I just mentioned to you with reversing aging in vitro was done with lentiviruses or with just regular transfection with DNA that had viral promoters, but no actual viruses. But yeah, I'm actually quite experienced at gene therapy. I've developed a lot of companies now use gene therapy therapies that I developed, including adenovirus and adeno-associated virus. Yeah, I definitely would like to talk. Definitely.
Yeah, I think, and you know, I have a special interest in Alzheimer's. My father had passed away with Alzheimer's and dementia.
Mine too.
And, you know, I read that the simplex virus can cross the blood-brain barrier. And so it would be interesting when you said telomerase helped in the mice, if you could do that with humans and somehow cross the blood-brain barrier using simplex or something like that, now you may be able to help reverse Alzheimer's or dementia.
Well, a lot of viruses pass the blood-brain barrier. There's papers saying that AAV9 does, but there's no papers that say the other ones don't. And actually, so we're using a different version that's actually proprietary right now that does cross the blood-brain barrier. But even though it does, our clinical studies, I've got a 200-page clinical protocol that I've written for treating Alzheimer's disease. Not only did my father die from Alzheimer's, but my younger brother died from Alzheimer's 3 months before my father did. Okay, so they both had the ApoE4 gene, and I don't, which is good. But the what was I going to say? I was going to say something about the gene therapy that you just alluded to, but I'm going. When I listen to the video, I'm going to remember what I was going to say. But the so gene therapy. Oh yeah. Even though they do get across the blood-brain barrier, intrathecal injections or pancreas, whatever that word is, directly into the brain is also very efficient at delivering to the brain. And in the case of like gene therapy, you probably only need the therapy once every 20 years. So it's not going to be a big deal to have a intrathecal injection or what's that word? Intraparenchymal? It's where you inject straight into the brain through the head. But I got it on my slides, but I can't remember the word. I show slides on ways of doing this. But the big problem with gene therapy is it's so expensive. Okay, so in our clinical studies, to treat one patient once costs $1 million.
Oh my.
Okay, that's out of my pocket. Okay, and so that's why Labella Gene Therapeutics was unable to treat their first patient, because they were never able to raise the money to treat one patient. That's
why when I did my gene therapy company, Actus, I did that in India because the cost in India was about 1/10 at that time, compared to the US. So doing some of these things may be better done outside the US because the costs are much lower.
And that's exactly what I recommend. In fact, I've spoken in India. I've been in Delhi, India, speaking a lot. I interact with a lot of people in India, but I've also been interacting with people in Colombia. I've spoken in hospitals. In fact, Labella Gene Therapeutics arranged to have their clinical studies done in Colombia, in Bogota, Colombia, and also Bucaramanga, Colombia, and also one other place in Colombia. But they could never get their acts together, and it just never happened. And I'm on the sideline. Okay, I'm doing research at the lab bench. I'm licensing these things to these companies that supposedly know what they're doing, and they don't get it done. You and I definitely have to talk. Okay,
Yeah.
I'm all for doing
I
it.
just gave you Bill's email address in Premium in Slack.
Good.
Great.
And
Here's a follow-up. Joanne asked some time ago, does the SOD gene protect against telomere degeneration?
yes, it does. And I don't like the word degeneration, but it's the free radicals do actually cause damage to what are called triplet Gs, so guanines in the DNA, and that causes, and so when the DNA gets repaired, it causes sometimes the telomere to be broken, and so it's called accelerated shorting. Things like SOD and other antioxidants will actually decrease the rate of that happening. So and you'll see in a slide coming up, I recommend antioxidants. Let's see, how do I get back to my screen?
You do in just a second. I want to show you something. There was a company that I helped some years back. I wonder if you've ever heard of them. I doubt it, but N3 has a product called the NanoV. Helps repair everyday cell damage caused by what you eat, what you breathe, what you're exposed to, and oxidative stress. Let's see. I'm going to see if I play this fast, if we get to something that. All right, it's too hard to.
So I started using the athletic training after a bone chip removal, as well as a microfracture surgery that I had. The machine was in there, I started using it as part of my recovery protocol, and I recovered from that in half time. Actually, I'm very fortunate since I've never been on the stable list, and haven't missed any time due to injuries. I'm very grateful for that. It has to do with my recovery, the way I train, and part of the way I recover is the NLG, and it is a huge reason why I've been at the field day in, day out. It can be for those athletes out there that one thing pertains to the next level. It's great for recovery, it's great for rehabbing, and it's great for just feeling good day in, day out without the fatigue that comes with a season and performing.
The more stress you put yourself under, the result of that, the more potential damage that's created. And so you must have a mechanism to protect yourself from that. And you drive.
Okay, so produces the same biological signal your body makes to repair cell damage brought on by free radicals, also known as ROS. Supplements the one your body makes. Have you heard of this? Does it seem?
I haven't heard. I do know that it's impossible to repair free radical damage. Okay, because the cell has no way of knowing what to repair it back to. So I think that's they're for the sake of layman audiences, they're just using wrong terminology here, but they're probably doing something that's actually removing oxidative damage proteins and things like that. The problem with repairing oxidative damaged DNA is that that's what causes the mutations, not the damage. Okay, is the repair, our repair systems are inefficient. And so when a cell tries to repair damage to DNA, it often puts the wrong base into place, and that's when the mutations occur.
Okay, I'm going to send you this site afterward. I love your
everything I see right there makes complete sense to me. This probably works. I mean, but I'm just saying that the semantics they use was probably geared towards the layman audience.
It certainly is.
Yeah, and everything looks good here. And I think what they're doing here is they're saying exactly what I'm saying is anything you can do to reduce your oxidative stress is good for you. Okay, to a point, because free radicals are needed by our macrophages and neutrophils to actually fight bacteria. They shoot free radicals at bacteria like we shoot machine guns in a war.
Yeah, I remember this was one of the more challenging assignments I had in terms of getting my head around understanding completely what was going on there.
I have a fun job. All I have to do is worry about how things happen, mechanisms. I get to spend all day just studying these things and reading papers and learning, but I don't get involved in the actual products and stuff like that. So I'll look at that stuff and find out exactly what they're doing. But everything you just showed me was 100% correct.
Jonathan wants to know how your work compares with David Sinclair's work.
Well, okay, so.
Okay.
I mean, it's like there's problems with me and David Sinclair. Okay.
Why don't we save that for offline then?
Yeah. Okay. Good.
You want to share your screen again?
Yeah. Let me look on it.
Do you have much more in terms of what you prepared?
I do, but I can go over it kind of fast. I thought I'd do whatever I've done so far in the first 5 minutes, but
yeah, we're not that kind of place.
Yeah, I know, and believe me, I enjoyed this better than anything else. This is the way I'd love to do all my things, but I'm looking at the clock and I've already been on for an hour and a half. So let me
Hour
just,
and a half is pretty much as long as we've ever gone, but
really? Well,
pretty
shoot,
much.
because I haven't even gotten to the main stuff I wanted to talk about. Let's see. So we reversed aging by every method of measurement.
You can see your screen if that's what you intend.
Oh, you don't see it? Okay, so I thought
You know.
I did hit the share button. Let me just go back and do it again.
Yes,
Oh, okay, so we're okay, good, because that's what I did last time too.
that's your email.
I know you don't want to see that, or I don't want you to see
do.
okay, so practically every disease we've ever heard of called, I call them telomeropathies because they're all caused by short telomeres. It plays a really big role in everything. The bottom line is bad things happen when telomeres get short. But this is the key thing I want to talk about is go-to-market partnership opportunities.
And I'm going to just color it a bit. Jan wrote, I'm going to read what she wrote because I'm amused by the way she wrote it. I've waited the entire time for him to tell us how his discovery will help us.
Okay.
Like we've talked a lot about the background and it's certainly helped us be grounded in it, but what can we do? And I suspect that this slide addresses that.
It does, and like especially the top three things on that list there, these are go-to-market partnership opportunities. The key is everything I talk about is also good for pets. But right now, since we are funded by people licensing things from us and marketing them, this is how to help us get our research done, is to either help connect us to somebody that might want to market one of these products. But these are all products that come by accident, by in our research that we discovered that then we just put on the shelf. And when I had investors, we were never allowed to do anything with them. And now, shoot, I've already licensed a bunch of these things. We survive right now on companies selling some of these products, but telomere length measurement is one of them. And this is where medical devices could come in handy. There's a lot of procedures out there right now to measure telomere length. And they're all awful. They're all terrible, but but people look at them as, hey, this is a way for us to make some money because people believe in telomere length. And so if we tell them we can measure your telomere length, people will pay us money, just like a fortune teller would tell you, I can read the palm of your hand and tell you how long you have to live. But none of these procedures work very well. Part of the reason is that we have so many different telomeres inside of our cells. They're all different lengths. And then like a lot of publications come out like this showing that telomere length is correlated with age, and there's a statistically significant score showing so. But because of my background in statistical theory, I know that statistical significance does not mean strong relationship. It just means.
I'm going to interrupt you for a minute. You've told us many times that you're a scientist, not a businessman, not a marketer. I am a marketer.
Yeah.
And I will say that this hour, the thing that I'm going to, remember at the very beginning of the talk, you and Liz presented 5 years ago.
Yeah.
And I've not recently watched the video. And as I recall, I remember, oh, the aglet story. I remember that and how they shortened, and that's bad. That's what I remember. This talk, you've taught us a great deal about, again, the ends. And when you get to 5, that's bad and you die. And you just said, yeah, people come to me and they say, yeah, help us lengthen our telomeres. I will say that what you've fed us so far is that if your telomeres were longer, good.
Yes.
And so they come to you and they say, hey, help us do good and lengthen our telomeres. And it sounds as though you're just about to go down a path of these poor misguided souls. They come to me, they say, lengthen our telomeres, like that's going to do anything. Now I'm confused.
I'm saying the ability to measure them is not very good. Okay.
Okay.
But lengthening them is good. And we know that from in vitro work, and we know it from using protocols. Let me go back to the second one from the bottom, where it says for research only. That's a really good method of measure, but it costs us $1,000 to measure one person's blood. Okay. Mm-hmm. So it's not something that's commercially available. But what I'm trying to say is that
I think we met at Daniel Kraft's event, I'm sure.
I don't know who that is.
Oh, okay. I was thinking that it was exponential medicine at the time, or it was Future Med at the time. Then it's exponential medicine. I don't know. I don't know how I found you then if it wasn't through him. But I think about how, like Moore's Law, how it was absurdly expensive to do anything back in the 60s. And now it's a fraction of a penny to fit all of that in this tiny little, is the thought that we just need somebody really fucking rich to do this so that we can get it to a point where everybody can have access to this.
Yeah, I mean, I even had Donald Trump before he was president offer to invest, but he wanted so much in return and wanted so much control. I turned him down.
That's surprising. Go on.
Yeah, Peter Nygaard, the guy who's just been all over in the news for sex trafficking. He's in jail now up in Canada. He did the same thing. And both of them wanted to take out loans on my behalf, and then wanted 80% control of the company. And I'd already been through that, been there, done that, with the investors. I want to get this research done. I don't want return on investment minded people trying to tell me to do something else that will bring quicker return on investment. That's why I'm not doing these products myself. I'm trying to find somebody else to take them on because I want to do research. I don't want to get involved in marketing and stuff like that. So telomere length measurement, it's making a lot of people are really good marketers, are making a lot of money doing it, but the data really looks like that, okay, which is crap. It's sure it's statistically significant, but it doesn't mean anything, okay? I've come up with a way to measure telomere length through long read DNA sequencing. Okay, the companies that are providing this right now, they can't do it because they can't figure out how to sequence all the way to the end. They don't know if they've reached the end or if the enzyme or whatever protocol.
Your point that you can, but it's cost prohibitive?
No, I got other things to do. I'm trying to come up with a cure for aging. I'm not trying to figure out a way to measure it. This is just something we figured out. We figured out their problem. We figured out how to identify the end of the telomere. I'm trying to talk to these companies right now. They seem like so busy trying to make all the money they're already making that they don't want to talk to me. So I'm looking for go-to-market partners to help us do this. Not only can we measure.
You're calling them up and saying, hey, I have the answer to how you can actually do this effectively. Like, look, we're really busy.
Yes.
Can you send us an email or something?
Exactly. They'll get back to me. I've sent and emails, I resend them and I say, what's going on? I haven't heard from you guys. I don't have time because I'm doing science to actually spend all the time that a business person might spend to do this. So I'm looking to you guys. This is why I want to talk to your conference. I'm looking for you guys for help here. Okay, I'm willing to share everything. I personally don't care if I ever make a penny off of curing aging. I just want to see aging get cured. I think it's the worst thing that ever hit this planet. But that's why I'm saying go to market partners. Does anybody have any idea? We have this incredible procedure that we can provide for somebody else to market if somebody would take it on. And I think we're going to learn a lot from it too, because it can identify which chromosome, not just all the telomeres. It tells us which chromosome the telomeres on that we're looking at. But then controlling telomere. This is, I think, where everybody wants to get to. Controlling telomere length is the problem. There's two ways. One is reducing the rate of telomere shortening. One is lengthening telomeres with telomerase.
Mm-hmm.
And the key to decreasing the rate of shortening is, we've already been through this. What?
Can I try to conceptualize through telomerase? Again, I was saying, is that something I would have as a pharmacological solution or inject it into my blood or what? How do I get telomerase?
Telomerase is too big. If you swallowed it or ate an animal that produces it, it's not going to help you whatsoever because it's got to get inside your cells and it can't get inside your cells. But first, let me just say it again.
Unless what?
Unless you get it to be produced inside the cells. Okay.
Okay, so I need to, so what can I do to produce it?
Let me get through this slide because that's the next slide after this. Okay, so reduce inflammation, oxidative stress will decrease the rate of shortening. These are the things that we've already talked about a little bit, antioxidants.
That are associated with good health.
These things will help go-to-market partners. We have a whole concoction of things that we could provide people, but the lengthening telomeres with telomerase. Okay, so we have nutraceuticals, we have pharmaceuticals that will induce our cells to produce telomerase. Okay. We also have gene delivery or gene therapy, as we were talking about before. The nutraceuticals and the pharmaceuticals, that's our major focus, especially the pharmaceuticals, only because I think that we're more likely to succeed with those than nutraceuticals. This is our system. Okay, we have all these departments in my company. This is why we have 10,000 square feet of space. We have high throughput screening using robots to be testing pharmaceuticals and natural products. We have this assay that we've developed called the TERT expression assay. Nobody else in the world has been able to come up with such an assay. We're the only ones that have this. We can identify drugs and natural products that produce telomerase. And that's a product right there too, just because even if a go-to-market partner wants to provide that to research labs to do this thing, that's available. But after our robots shown in the bottom here, go through testing 4,000 different things. They're sent back to these different departments to redesign them and stuff like that. And it's back and forth. And through this process, we've now discovered thousands of telomerase inducers. Okay, but telomere shortening and lengthening is like a tug of war. Okay, it's just that without telomere lengthening, you only have a one-sided tug of war, so the telomeres are always shortening. When we come up with a telomerase inducer that adds somebody to the other side to lengthen, but in everything we discovered so far, doesn't have enough on this side to win the tug of war. The tug of war still shortens. Okay, so we estimated years ago that if our investors didn't interfere with our research and we could have continued doing what we're doing, we could have had a drug, a pharmaceutical or nutraceutical, that would lengthen telomeres in 1 to 3 years if we had continued being able to get our $1 million a month funding. But that all ended with the global financial crisis of 2008. We ended up having to resort to the go-to-market partners as a way of bringing in funding, and we've never gotten back up to that. But if we could find somebody that could provide us with $1 million a month or some go-to-market partnership that we could have a pill that you could swallow that will reduce enough telomerase to lengthen telomeres and reverse aging, or at least allow us to ask the question, does it reverse aging? Does it reverse decline in health? Does it reverse Alzheimer's and all those other diseases?
All right, what I'd like to propose, we're at 90 minutes, and not just because 90 minutes is about as long as I can pay attention to anything. Why don't we set up maybe an hour next week?
Okay.
You and me, and Sanjeev, if you want to come, if anyone else wants to write me a note now saying that you want to, and we could start basically here.
I'm going to
start at this end part where we're talking about either shortening or lengthening, pardon me, reducing the rate of shortening or lengthening. Let us get our heads around that so that we can talk about that. Yeah.
Okay. This page is probably the best page to leave off at. This is just from our website. If you just go to www.sierrasciences.com/go-to-market-partners, there's a yellow button. You click on that, that'll take you to a document that describes everything. Okay. People might want to read that before we do our next talk, so that maybe at our next talk, we can focus just on the questions and answers.
Yeah. This is the 56th one of these that we've done. It's the first time that I was like, at 90 minutes, like, okay, we need to talk more so I can.
I never thought that this would take more than half an hour to present, but
fool.
I'm glad. I like the dialogue. A lot of people hate being interrupted when they're talking. I'm the exact opposite, and that's probably my answers probably took up more time than your questions.
No, you know, the reason I enjoy this format so much, it comes very naturally to me, and I don't have to prepare anything. And I do that on purpose because I want the questions that I have, which are reasonable for the most part, to be as uninformed as everyone else who has absolutely done no preparation to come to this talk. And their questions are probably going to be like, okay, what did that mean? What did that mean? And if I'm any smarter than kind of that level of preparation, I won't ask that and I'm not able to represent for them. But it's fluid and it's a conversation. And I think that makes it more digestible.
I am so open to any advice you have. I think this is the greatest invention, medical invention of all time. What can I do to make it actually get going? And I need you guys to help. Okay, so let's do it.
A fitting way to end the 2020 MDG Premium Series, because the next 2 Fridays are Christmas and New Year's. And yes, we're going to take off for those. Our next meeting will be on January 8th. And I'll make it available to everybody. I have 3 people raising their hands saying they want to join us next week. I'm thinking maybe Tuesday, Wednesday for an hour. We'll find a time, if that makes sense.
Okay, good. And if anybody who doesn't have any background joins, how will they know this stuff in advance? If there was some way you could.
I'll still have this metric of being able to, you know, filter a bit, and I can kind of keep us
good
going rather smoothly. So
thank you.
Thank you.
Thank you. Okay, so
you,
all right. Is that it?
See you next week.
See you next week. All right. Thanks.